Hello, we are team number 4, and our experiment's objective is basically to identify and analyze the different pigments that are present in leaves, such as spinach leaves, and how their mixtures are able to stick up onto the paper chromatography. The materials we used for this experiment were chromatography papers, scissors enabled to cut the paper, mustard and pestle, spinach leaves, acetone and ethanol in order to make the mixture, a ruler, a pencil, and a pipette.
For our experiment, we grabbed around 3 spinach leaves and we cut them up into really small pieces so that we could fit them well into a mortar. Then we started grinding them up into a sort of paste. And to that paste, we added about 10 drops of acetone to make sure that the pigments that had been released would settle down. We let those settle down like fully at the bottom for some time while we prepared our chromatography paper. And we made sure that the point in our chromatography paper measured around 1.5 centimeters. Then we grabbed a pipette and sucked up the pigments that had been released from our paste and put them along the line in our chromatography paper. We then placed that paper on a beaker with 10 milliliters of acetone. For 15 minutes, we let that rest, and then we measured the retention factor. We then repeated the experiment twice with ethanol and once with a different colored plant, which was purple, but with acetone.
For the chromatography paper of spinach, we were able to see this on acetone and this on ethanol, and this for the purple plant. The different pigments observed within the spinach were, in acetone we were able to visualize the chlorophyll B and the carotenoids. In the ethanol we were able to visualize the chlorophyll B, carotenoids, and a little bit of xanthophyll. And in the purple we were able to visualize the chlorophyll B and the anthocyanin, which is the one that gives the reddish and purple pigment to plants. While using both acetone and ethanol, we were able to conclude that the ethanol gives us a higher resolution in the chromatography paper, giving us a more wide variety of color spectrum and pigment separation than the acetone did. The photosynthetic pigments shown in the chromatography paper help us conclude the absorption and the action rate that each of these plants have by knowing the color of the plant and the pigments that it used, meaning that the color of the plant is a pigment that was shown, that it reflected light, but the ones that were not shown in the white eye on the plant were the ones that actually absorbed the light and that were used mostly for photosynthesis action, thus making the chromatography paper useful for us to identify the absorption rate and the action rate to a medium level of the plant that we were taking these pigments from.
So from this experimental activity we can conclude and we can see how the chromatography experiment functions. And we can actually see the difference between pigments. For example, in the first two experiments we used a green plant, and also we could see the reaction of these pigments between two different substances.